Hello viewers, welcome to today's live. In this live of today, I will be reading chapter number 2 of class ninth from the book Exploration NCERT, and the name of the chapter is Cell, the Building Block of Life. Please confirm me for the audio. Is my voice clear to everyone so that we can go ahead? Is it audible to everyone? Is my voice clear so that we can start? So this is chapter number 2, which is actually the first chapter of bio in your book. And this is going to be a lengthy chapter because if you compare it with past year, not many activities are being added in this chapter now. So in case while hearing the audio throughout, in case you find that any topic which is difficult for you and you are not able to understand, you can comment me. I'll get back to you by recording a separate video and then you can watch it for clarifying your concepts. This is actually the audio which will help you how to pronounce these words, because there might be certain terms in the chapter which you will not feel easy at. So without further ado, let's begin. Yes, it's very much clear. Even I can see it in my window. In case of any doubt, you can just comment underneath this video. So, it is widely accepted amongst the scientific community that life originated in water. Some researchers believe that life may have originated in small water pools with changing environmental conditions rather than in the oceans. Hot springs are examples of such environments. In India, the hot springs of Fuga Valley in Ladakh maintain very high temperatures nearly at the boiling point of water even in a cold climate. These environmental conditions seem to be similar to those on the early Earth about 3.5 billion years ago. The organisms living in these hot springs are mostly heat-loving bacteria called thermophiles, which are unicellular. Scientists from the Birbal Sahani Institute of Paleosciences, Lucknow, studied these hot springs and found that calcium carbonate formed rapidly around them. These deposits may have protected early organic molecules from harmful radiation and extreme conditions, and they may have also helped in the formation of the first protective membrane, the barrier that defines a cell. All living organisms are made up of cells. The cells represents the basic level at which life exists. Some organisms such as bacteria or yeast consist of only one cell or unicellular, while others like plants, fishes, birds or humans are made up of millions of cells, multicellular, that work together. A group of similar cells performing similar functions forms tissues. Different tissues are organized to form an organ, and several organs work together to form organ systems, such as nasal pores, nasal cavity, trachea, and lungs form respiratory systems. Even when cells are organized into tissues, organs, and organ systems, the cell remains the fundamental unit of structure and function in all living organisms. This makes us wonder how such tiny cells perform so many different activities. What are the different components of a cell? How do cells in our body communicate with each other? Do cells live forever or do they die? In this chapter, we will explore the answers to all these questions and we enter the fascinating world of cells. So, how to study cells? What do we call the ability of the human eye to see two very close objects as separate and distinct? Imagine two tiny dots drawn on a piece of a paper. As the dots are moved closer, there comes a point at which they can no longer be seen as separate. When viewed from about 25 centimeter, which is the near point of the human eye, 2 points separated by about zero point 0.1 millimeter and can be seen as distinct. Otherwise, they appear as a single point. This is called the limit of resolution of the human eye, which is zero point 0.1 millimeter. A cell is usually too small to be seen by the unaided eye. This raises an important question. How have cell biologists studied the structure and function of cells that are much smaller than the limit of resolution of the human eye? You have learned about convex lens. A convex lens, or a combination of lenses, that is an objective lens and an eyepiece, are used for the magnification of an object to make it appear larger. Robert Hooke was the first person to observe a cell in 1665 using a self-designed microscope capable of about 200 to 300x magnification. While examining a thin slice of cork, he observed small boxes of compartments and named them cells. In school laboratories, light microscopes are used to observe objects using different objective lenses, which could be 10x or 40x, to achieve better magnification and resolution under visible light. Explore different parts of a microscope in your school laboratory and use it to observe fine structures of various materials. Under the microscope, you will see a magnified image of an object. Can you estimate its actual size? So here we come on to the next activity 2.1, in which we will estimate the size of a cell. Take a transparent ruler with millimeter or markings. Place the ruler on the stage of the microscope, focus on it using the adjustment knob, and observe the diameter of the circular field of view through the eyepiece and measure it in millimeter. Convert the diameter from to micrometer. Here is the symbol for micrometer. Suppose the diameter of the visible field is 5 millimeter, meaning 5 into 1000, that becomes 5000 micrometer. Remove the ruler and place an onion peel slide on the stage of a microscope. Focus on the slide and count the number of cells present along the diameter of the field of view in one straight line. Estimate the real size of the cell using the formula. Unit conversion is 1 millimeter is equals to 1000 micrometer, and the formula that you need to put is estimated size of the onion peel cell will be equal to the diameter of the visible field in micrometer divided by the number of cells along the diameter. Suppose 25 cells are seen along the diameter. In that case, the size of one onion cell will be equal to 5000 divided by 25 micrometer, that gives you 200 micrometer. If the estimated size of an onion peel cell is 200 micrometer, how much does a light microscope magnify this cell? The total magnification of a microscope depends on its magnifying power of the eyepiece and the objective lens. If both the eyepiece and the objective lens have the magnifying power of 10x, then the total magnification will become 100x. This means that a cell with an estimated size of 200 micrometer will appear 100 times larger. Thus, a microscope allows us to see very small structures clearly and is an essential tool for studying the cell structure. Over the years, scientists have improved the microscope by improving its three main features: resolution, which is a measure of clarity; contrast, the difference in brightness between various parts of an object; and magnification. These improvements have turned microscope into a powerful tool for studying the cells. You can see a picture of the lower surface of colocasia leaf observed under the scanning electron microscope. which is also abbreviated as SEM. We come on to the next topic, structure of a cell. You have learned that cells are organized into specialized tissues and organs and collectively perform specific function. For these cells to function as units, they must be able to interact with one another and with their surroundings. These interaction occurs at the cell boundary, where substances move between the cells and their external environment. Even single-celled organisms exchange materials and respond to their environment through their cell membrane. Cell membrane, the universal feature of a cell. The cell membrane is a thin boundary that surrounds a cell and protects its content. It defines the individuality of a cell and is also called the plasma membrane. The cell membrane is a selectively permeable, which means it allows some substances to pass through it while blocking the others. You have learned how oxygen and carbon dioxide move across the membranes of alveoli in the lungs. How does the structure of the cell membrane in the cells of alveoli control the movement of substances across it? There is an activity 2.2, wherein you will be told about osmosis. So with the help of a kitchen knife, carefully cut a potato into 2 pieces of roughly equal size. Measure and record the initial weight of both the pieces using a weighing balance. Put 1 piece of the potato in beaker A with plain water. Put the another piece of the potato in beaker B with 20% salt or sugar solution. This you have already understood, how to prepare 20% of the salt or sugar solution. Please write it in the comments how to do it. Next, leave them undisturbed for about an hour or until you notice a visible change in the size of the pieces. Measure and record the final weight of each piece. Calculate the difference between the initial and their final weights. What do you observe? You may observe that beaker A, the potato swells, whereas in the beaker B, it is going to shrink. What do you infer? What do you expect in terms of changes in their weight? The weight of the potato piece in beaker A has increased, while the weight of the potato piece in beaker B has decreased. This happens because the cell membrane allows water to move in and out of the cell, but not the sugar or salt molecules. Water moves from an area with more water and less solute or dilute solution to an area with less water and more solute or concentrated solution, until the concentrations in the two areas become equal. This movement of water through a selectively permeable membrane is known as osmosis. You have learned in activity 7.8, which is dye spreading in water, and 7.9, fragrance spreading in water, in grade 8, that particles of matter intermix due to the difference in their concentration, also called a concentration gradient. Diffusion is the net movement of particles from a region of higher to lower concentration, which occurs even without membrane. Osmosis is the diffusion of water across a selectively permeable membrane. In plants, water from the soil enters the root cells by the process of osmosis. So here is another activity which is showing you the different types of solution. The names of the solution are isotonic, hypotonic and hypertonic. So a cell is kept in salt or sugar solution of different concentrations. So isotonic solution is a one in which the salt concentration of extracellular medium is equal to the solute concentration of intracellular medium. Hypotonic is a one in which solute concentration of extracellular medium is lesser than the solute concentration of intracellular medium, whereas hypertonic solution is a one in which solute concentration of the extracellular medium is greater than the solute concentration of the intracellular medium. Now here we come on to the fluid mosaic model, which actually described the structure of the plasma membrane. It was given by Singer and Nicholson. Singer and Nicholson are the names of the scientists who gave this model. So structurally, the cell membrane is extremely thin, about 7 to 10 nanometers thick. It is made up of lipids, which are also known as fats and proteins. The fluid mosaic model explains its structure. The membrane has a lipid bilayer, two layers of special fat molecules with water attracting heads outward and water repelling tails inward, with proteins embedded in them. The molecules can move sideways, flip, and rotate within the membrane, hence, it is known as fluid. Proteins in the membrane act like gatekeepers in helping substances pass through. Since the molecules are arranged like tiles in a mosaic, it is called as the mosaic model. All living cells communicate with their surroundings and their neighboring cells through the cell membrane. However, cells of plant, fungi, and bacteria have an additional layer around the cell membrane called the cell wall. What do you think is the necessity of the cell wall in these cells? Activity 2.3. First step is prepare temporary slides of a thin peel of onion leaf or a royo cradle lily leaf and mount it with safranin using the cover slip to observe plant cells under a microscope. The second step is similarly prepare a temporary slide of the cheek cells by gently scraping the inner side of your cheek with a cotton swab or the blunt end of a toothpick. Third one, spread the cheek cells on a clean glass slide. And fourth one, add a drop of water followed by a few drops of methylene blue stain and carefully place a cover slip. Observe both the slide under a microscope. What do you observe? Onion peel cells or royo leaf peel cells are box shaped and regularly arranged, whereas cheek cells are irregularly arranged. Why do you think this difference exists? Prepare two slides of a royo leaf peel and human cheek cells again and put 20% of the sugar solution on them. I think it is easy for you to understand once you are asked to prepare 20% sugar solution. That means if it is mass by volume, then you need to take 20 gram of the sugar and you need to dissolve it in 100 ml of the water. Okay, observe them under a microscope after half an hour. What do you observe? You must have observed that the boundaries of the plant cells remains the same, but their inner content shrinks and the shape or the space between the inner and the outer boundaries increases. You may observe that the cheek cells, on the other hand, have shrunk considerably. Cell wall, the outer covering of the cells. In general, plants cannot move from place to place, so they need a rigid structure to withstand environmental stresses like wind and rain. Therefore, plant cells have an additional covering outside the cell membrane called the cell wall. The cell wall also helps leaves and flowers remain firm and maintain their shapes and help plants stand or stay upright. Although rigid, the cell wall is permeable, which means water and some dissolved minerals can pass through it. Along with the selective permeability of the cell membrane, the permeability of the cell wall helps plant roots absorb water and nutrients from the soil. When we place a royo leaf or an onion peel in a concentrated sugar solution, the plant cells lose water due to osmosis. However, the cells do not shrink in size because their rigid cell wall maintains their shape. The inner content of a cell shrinks as the cell membrane pulls away from the cell wall. This shows that the cell wall helps plant cells remain firm in their original shape. Animal cells such as cheek cells do not have a cell wall. Therefore, when placed in a concentrated sugar solution, they lose water and they shrink. Without a rigid cell wall, animal cells can change shape considerably. This cellular flexibility supports the overall movement and functioning of the animal tissues. The plant cell wall is primarily made up of cellulose, a type of carbohydrate formed by many glucose units linked together. Cellulose in our diet acts as a roughage, helping in digestion. Some microorganisms like fungi and bacteria also have a cell wall to provide protection and structural support to their cells. The cell interior, a coordinated working system. It is composed of three parts: a selectively permeable membrane called the plasma membrane, a semi-fluid or a jelly-like substance called the cytoplasm, and a prominent nucleus. In addition to the nucleus, the cytoplasm contains several subcellular components called organelles, along with other substances present in it, most of which are only visible with an electron microscope. So there is an activity in which you have to study the two types of cell. The first step is study the given diagrams of a bacterial cell, a plant cell, and an animal cell. Observe the different structures present in each of them, and you need to record your observations. A bacterial cell lacks a well-defined nucleus and membrane-bound organelles. Organelles surrounded by their own membrane are known as membrane-bound. Such cells are called prokaryotic, wherein pro means primitive and karyotic or karyon means the nucleus. In prokaryotic cells, most cellular activities take place directly in the cytoplasm. In contrast, plant and animal cells have a well-defined nucleus and several membrane-bound organelles. Such cells are known as eukaryotic cells. Eu always means true and karyon always means nucleus. More details of the prokaryotic and the eukaryotic are given here in this table 2.2. So a nucleus well defined is present in the case of eukaryotic but not in the prokaryotic. That is why it is written here primitive nucleus is present in prokaryotic, because the nucleus is well developed in eukaryotic. Diameter of prokaryotic is smaller. It is just 1 to 10 micrometer, whereas for eukaryotic it is 10 to 100 micrometer. Number of cells in prokaryotic, they are generally unicellular or single celled, whereas eukaryotic can be unicellular or multicellular. Membrane bound organelles are absent in prokaryotic and they are present in eukaryotic. Membrane bound nucleus is absent in prokaryotic and present in eukaryotic. One question of differentiate is 100% sure will come to you from this part. So why do eukaryotic cells need these organelles? Eukaryotic cells carry out various life processes in different cell organelles independently at the same time. Cell organelles help in building new materials, removing waste, and providing energy to the cell. They work together to perform all functions of a cell. In other words, a cell is just like a tiny living factory with each of its parts doing specific job. Let us explore these specialized structures present within a cell. Nucleus, which is a house of coded instructions. The nucleus has a double layered covering called the nuclear membrane, which has pores that allow the transfer of material between the nucleus and the cytoplasm. The nucleolus is the dense round body in the nucleus where synthesis of ribosomal subunits takes place. These subunits then exit the nucleus to cytoplasm where one large and one small subunits assemble to form the ribosome. The nucleus contains the chromosomes, which are visible as rod-shaped structures only when the cell is about to divide. Chromosomes contain information for inheritance of characters from parents to the next generation in the form of DNA, which is deoxyribonucleic acid molecules. Chromosomes are composed of DNA and specific proteins. DNA molecules contain the genetic information. The functional segments of DNA are called genes. In a non-dividing cell, this DNA is present as part of chromatin material. Chromatin material is visible as an entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into the chromosomes. Prokaryotic cells do not have a well-defined nucleus. Their DNA is present as a single circular molecule associated with specific proteins, and the region containing this genetic material is known as nucleoid. The next cell organelle is ribosome, which are known as the protein factories. These are the tiny structures may be present either freely in the cytoplasm or attached to the endoplasmic reticulum. Ribosomes are the sites of protein synthesis. Endoplasmic reticulum is the manufacturing factory. The ER is a large organelle that spreads like a network within the cytoplasm of the cell. The ER is continuous with the outer membrane of the nuclear envelope. The ER plays a key role in the synthesis and transport of proteins or fats and some hormones in some of the specialized cell. The structure of the ER in a cell varies depending on its function. There are two types of ER. Rough endoplasmic reticulum or RER. It looks rough under the electron microscope because it has ribosomes attached to its surface and is mainly involved in protein synthesis and protein secretion, for example, in the gland cells such as pancreatic cells. Smooth endoplasmic reticulum or SER do not have ribosomes on its surface and therefore looks smooth. It is involved in the synthesis and storage of fats and the hormones. Golgi apparatus, the packaging and the shipping centers. If you carefully look at the diagram of a cell, you will notice stalks of flattened sac-like structures. Together, these stalks forms the Golgi apparatus. It is functionally linked to the ER, the cell membrane, and the other cell organelles. The Golgi apparatus acts like the cell's post office. It modifies, sorts, and packages the proteins and or lipids into vesicles for transport, secretion, or lysosome formation. Next is lysosomes, the cleanup system. Cells produce waste materials and damage worn-out organelles during their activities. How does the cell prevent these wastes from accumulating inside it? Lysosomes are single membrane bound sacs filled with enzymes which can break down unwanted proteins, carbohydrates, fats, and even the damaged parts of the cell, keeping it clean and healthy. The products formed by the breakdown are released into the cytoplasm where they may be reused in other cellular processes. Mitochondria. The powerhouse of the cell. Mitochondria is called as the powerhouse of the cell because they supply the energy needed for most cellular activities. Each mitochondrion is surrounded by two membranes. The outer membrane, which is smooth and porous. The inner membrane is folded into finger-like projections called cristae, which increase the surface area for chemical reactions and facilitate energy production. In mitochondria, the glucose and other molecules are broken down to release energy during a process called cellular respiration. You have already studied about it. The energy released is stored in the form of a molecule called adenosine triphosphate or ATP, which acts as the energy currency and is used for most of the cellular activities. Plastids are the center for food synthesis in the plant cells and beyond. You have learned that mitochondria provide energy to the cell that comes from food. Animals can obtain food from their surroundings. However, plants synthesize food in the presence of sunlight. But where do plants synthesize their food and obtain energy for cellular activities? Plants use special organelles called plastids for food synthesis and storage. As you have learned, plants prepare their food by the process of photosynthesis in the presence of sunlight. A green pigment called chlorophyll, which is present inside the chloroplast, absorbs the sunlight. Chloroplasts are double membranous bound organelles like mitochondria. Inside the chloroplast, there is a semi-fluid substance which is called stroma. Within the stroma are dish-shaped membrane structures that contain the chlorophyll. Light energy is absorbed by them during photosynthesis. The sugars synthesized in this process are stored in stroma along with the starch granules. Mitochondria and plastids have some features that are similar to certain bacteria. For example, they both have their own DNA and ribosomes, and thus they can make some of their own proteins. These characteristics suggest that both mitochondria and plastids share an evolutionary history with these single-celled organisms. Are there any other plastids in plant cells that contain any pigments other than the green pigments? How do flowers, fruits, and vegetables acquire various colors? In flowers, petal, and fruits, plastids contain pigment other than chlorophyll. These plastids are called chromoplasts, which has been derived from a Greek word chroma means color. Their pigments may be yellow, orange, or red, are the source of bright colors in such flowers and fruits. Such bright colors help in attracting the pollinators for pollination and fruit-eating animals that help in the seed dispersal. Some plastids lack the pigments and are thus colorless. They are called leucoplasts, derived from the Greek word leukos, which means white. Leucoplasts store food materials such as starch, oil, or proteins and are classified based on the type of the food they store. For example, some leucoplasts in potato and taro, which is actually colocasia or arbi cells, stores the starch. Vacuoles are the organelles of storage and support. Plastids such as chloroplast help plant produce food and temporarily store it. Other plastids such as leucoplasts help in storing the food, but where are water, minerals, and the waste materials stored in the cell? Why do plants look wilted when they do not get enough water? In a mature plant cell, there is usually one large central vacuole surrounded by a single selectively permeable membrane. The vacuole is filled with a watery fluid which is known as cell sap. The vacuole stores the water and the minerals, sugars and the waste material. By storing large amount of water, the vacuole helps maintaining the pressure inside the cell, which keeps a plant cell actually firm. When a plant does not get enough water, the vacuole loses water and the cell becomes less firm and the plant gets wilted. In animal cells, vacuoles are sometimes present. Although they are not as large as plant vacuoles, they help in temporarily storage of the materials. So when you get a small cut on your skin, it heals after few days. When hair falls out, new hair grows back. How does this happen? It happens because cells in our body can grow and divide to replace the old, dead or damaged cells. When our body grows, it is not just because cells get bigger. Cells can grow up to a certain size, but growth happens because cells divide to form new cells. Let us study freshly growing root tips of an onion to understand how cells divide. Let us enhance our skills. Take a jar and fill up with plain water. Now place onion bulb over the jar in such a way that its base bearing roots is immersed in the water. Leave the setup for about 5-6 days and observe. Do you observe the roots growing? Cut 2 to 3 centimeters of the freshly grown root and transfer them to freshly prepared aceto alcohol, which is actually the mixture of glacial acetic acid and ethanol in the ratio 1:3. Keep the root tips in aceto alcohol for about 24 hours and then transfer them to 70% ethanol for preservation. Take 1 or 2 preserved roots, wash them in water and then pack them on a clean slide. Place them on a clean slide. Put 1 drop of dilute HCl or hydrochloric acid on the root tips to soften the tissue. Rinse the roots after 10 to 15 minutes. Then add 2-3 drops of acetocarmine stain on them. Acetocarmine is a coloring agent which is basically used to color the chromosomes. Leave the slide for about 5 to 10 minutes and then gently warm it with caution under the spirit lamp. Cut the tip portion of the root and the slide and put the cover slip. Gently squash the cover slip with your thumb to spread the cells on the slide. Observe the slide under the microscope. What do you observe? Do you observe the cells of the onion root tip? Are they similar in structure? Do you find any structural differences in these cells? If yes, why is it so? This is because the cells of a growing tip of root of onion divide continuously. This process is called cell division, shows various stages of cell division. Therefore, these cells exhibit different structures corresponding to different stages of cell division. Can you identify which stage comes first during the cell division? Every day, an estimated hundreds of billions of cells in our body are replaced, which is almost 1% of the total number of the cells in our body. Both prokaryotic and eukaryotic cells divide, but eukaryotic divide in more controlled and orderly manner by a process called cell cycle. You will learn about the stages of cell cycle in the higher grades. Now we come on to the next topic, which is cell division. Cell division is a process by which new cells are formed from the pre-existing cells. It allows living organisms to grow, repair damaged tissues, and reproduce. Some cells, such as skin cells, divide continuously to replace cells that are lost regularly. There are 2 major types of cell division, mitosis and meiosis. Mitosis is important for normal growth, repair, maintenance, and asexual reproduction, while meiosis is important for sexual reproduction for creation of genetic diversity. Mitosis. Every human begins life as a single fertilized egg. This one cell divides repeatedly to form trillions of cells in the body. Cells increase in number through mitosis, which is the most common type of the cell division. Mitosis produces two genetically identical daughter cells from one parent cell. Each new cell gets the same DNA and the same number of chromosomes as the parent DNA. This ensures that the genetic information is largely maintained across the blood or body cells. Meiosis is a type of cell division that produces gametes and occurs only in the cells of the reproductive organs. Gametes produced for sexual reproduction create variations and diversity among the living organisms. Therefore, children resemble their parents but are not exactly same. In animals, including humans, meiosis occurs only in the cells of the testes of male to produce the sperm and ovaries of female to produce the eggs. In plants, meiosis occurs in the anther to produce pollen grains and the ovaries, which are actually the female parts, to produce the egg cells. In meiosis, the parent cell divides twice, one after the other, to form four daughter cells. During the first division, the cells divide into two daughter cells and the number of chromosomes in each of the daughter cell is reduced to half. The second division is similar to mitosis, where each daughter cell divides into two, thus forming four daughter cells with half the number of chromosomes. As a result, each gamete has half the number of DNA compared to the parent cell. During fertilization, when gametes from two different individuals combine, the original chromosome number is restored. You will be studying the details about it in chapter number 11. The processes of mitosis and meiosis must occur in a proper and a controlled manner. What happens if meiosis and mitosis does not happen properly? If there is any error in this process, it can lead to various problems in the body of the organism. Errors in mitosis lead to uncontrolled cell division. This can lead in the formation of tumors and abnormal number of chromosomes in the body cells. Errors in meiosis may result in the genetic disorders, which may be associated with developmental problems or distinct physical features. Faulty meiosis may also cause early pregnancy loss or reduce fertility. Cell theory, the unifying principle of biology. An important observation about living organisms is that all organisms are made up of cells. In 1838, a German botanist named Matthias Schleiden reported that all plants are made up of the cells. In 1839, German zoologist Theodor Schwann found that all animals are also made up of the cells. Later in 1855, a German scientist named Rudolf Virchow further expanded the cell theory by stating that new cells are formed only from the pre-existing cells. Together, their work led to the formulation of the cell theory, which has various postulates. And the first point is all living organisms are made up of one or more cells, and the cell is the basic unit of structure and function in living beings. All cells arise from the pre-existing cells. This unifies all biology from bacteria to humans and explains life continuity through the cell division. Do cells grow and reproduce forever? Cells grow and divide in a controlled way, stay in the right place, carry out their functions, and eventually die when they no longer needed. Dead cells are replaced by new cells that carry out the same function. Thus, every cell has a definite lifespan. If cells do not die when they should, or if they die too early, problems can arise in the body. In many animal cells, cell division usually stops when the cells comes in contact with the neighboring cells. This process is known as contact inhibition. However, cancerous cells lose this control and keep dividing uncontrollably, leading to the formation of tumors. Plant cells grow differently due to their rigid cell wall. Plant cells do not show contact inhibition and follow a different pattern of growth. Even though cells are extremely tiny, they perform remarkable functions. They produce energy, synthesize and secrete substances, divide to form new cells, and work together to maintain the proper functioning of the body. Every living being, from a tiny bacterium to a giant tree, is made up of the cells. You are one of the product of these tiny hardworking cells. So with this, we come to an end of the reading of chapter 2, Cell, the Building Block of Life. In case you are having problem in understanding any of the concept that has been taught to you in this chapter, you can comment me beneath this video and I'll get back to you with a video and will definitely specially mentioning your name. Stay connected. I'll be back with chapter 3 tomorrow.